While genes known as transcription factors, whose proteins bind to DNA and turn on genes, thus allowing genes to be regulated, while these are known in bacteria, eukaryotes developed more. Animals developed still more, metazoan animals even more, and the increasing number of these transcription factor genes aided in increasing complexity as animals evolved. There are shared genetic pathways using these transcription factors. So for example, in Drosophila, there are a number of homeodomain transcription factors, a subset of the helix-turn-helix family of transcription factors, which cause brain cells and eye cells in the embryo to develop a specific fate. So these are on-off switches helping these cells to become cells of the eye and to specialize. The same transcription factors have homologs in human embryos which aid human embryonic cells develop a brain fate or an eye fate. And so, shared genetic mechanisms indicate that the last common ancestor of humans and flies possessed homeodomain transcription factors which were crucial in eye development. These homeodomain transcription factors developed a subset known as the Hox genes, which occur in a tandem set. In other words, there is one gene located physically next to another gene, next to another gene, and it was duplications in tandem, so that after one gene accidentally became two, that the two lay adjacent to each other. These Hox clusters are fascinating because not only are they critical in developing the plan of a body as they are expressed along a body axis, the order of the genes in the cluster dictates the order of the expression of the genes along this axis in body patterning, whether it be along the anterior-posterior axis or along a limb or along the gut. And so their physical position in the cluster helps them map where they are expressed in the body. These Hox clusters were crucial in the development of complex animal body plans, and thus they can be traced throughout animal evolution. They were first discovered in fruit flies with the observation that certain genes, when mutated, caused a part of the fly to develop in an inappropriate place. So for example, in this fly here, the antennae have developed into legs. And so the mutation is that there is an on-off switch for leg development, and it is being expressed in the wrong part of the body. And because of this, the cells which should become antennae develop into legs instead. It was then discovered that humans have homologs of these transcription factors, and that the Hox clusters are crucial not only for giving a fly embryo its body plan, but human embryos its body plan. Now, because there have been duplications in genes since the last common ancestor of flies and humans, flies possess some genes which humans do not, since duplications occurred after the last common ancestor. And vertebrates famously underwent 2 genome duplications, meaning that the one ancestral Hox cluster and its neighboring clusters called para-Hox and NK clusters, they were duplicated once so that primitive fish had 2 such sets of clusters, and then duplicated again to make 4. And so humans have 4 Hox clusters inherited from an ancestral single Hox cluster. Some of these genes are no longer functional, having been mutated, but these Hox clusters are crucial in human development as human embryos develop a body plan.